Welcome everyone. Today we are going to learn about lipid rafts in the plasma membrane. So today's topic is lipid rafts. Now plasma membrane can be seen as the ocean of lipid molecules because I have mentioned that in the plasma membrane there is major three category of lipid molecules present: phosphoglycerides, sphingolipids and cholesterol. Now the question is whether these lipid molecules are well organized or well ordered, or they are just randomly mixed with each other. Now the first hint comes when scientists extract lipid molecules from plasma membrane. Okay, they use non-ionic detergent to extract the lipid molecules. One example is Triton X100. One example of non-ionic detergent, Triton X100. So when scientists extract lipid molecules from plasma membrane with the help of non-ionic detergent, Triton X100, they found that after the extraction, some lipid molecules remain in the plasma membrane. Some lipids remain in the plasma membrane. And when they analyze the lipid molecules, they find that these lipids are predominantly sphingomyelin and cholesterol. Now here from the journey of lipid rafts uncovered. So they found that these two species of lipid molecules remain after the extraction in the plasma membrane. And these two lipid molecules we know can found in the more ordered and less fluid bilayer. So these two lipid molecules can be found in more ordered and less fluid bilayer. So from this hint, scientists hypothesized that this kind of lipid molecules can come together and can bind very intactly and make microdomain. So they hypothesized that there may be some microdomain present, in the plasma membrane that they call lipid raft. Okay, so this is the first hint from the experiment. Now scientists perform another experiment by which another hint comes that demarcate whether lipid rafts are present or not. In these situations, scientists made a giant liposome. Now what is liposome? I have made a separate video on liposome. Liposome is actually artificial lipid bilayer. It is very similar with the plasma membrane, but in case of natural plasma membrane that is present in the cell, the plasma membrane contains lipids and proteins. But in case of liposome, it is the only lipid bilayer that are actually made in an aqueous solutions. So liposome is a lipid bilayer that can mimic plasma membrane. So for research purpose, membrane research, liposomes are actually taken. So scientists also taken giant liposomes. Now they actually make these liposomes with the help of two lipid molecules, lipid species. One is phosphatidylcholine. I have already mentioned, if you have watched my previous video, you know phosphatidylcholine is a group of phosphoglycerides. So they phosphatidylcholine and sphingomyelin. Just by taking two species of lipid molecules and in a ratio of 1:1, with the ratio of 1:1, they actually made the giant liposome. But in the giant liposome, there is a speciality in this lipid composition, that is, they have uniform bilayer. That means there is no like this ordered structure. There is no organizations or there is no order, just uniform bilayer. Okay. But when they introduce another lipid molecules, like in this giant liposome, they take phosphatidylcholine, sphingomyelin, additionally cholesterol. They just add cholesterol with a ratio of 1:1:1. They found that now the liposome contains domains or small domains.
As you can see, this is the structure of giant liposome. And the first structure is actually found when phosphatidylcholine and sphingomyelin are taken 1:1 ratio. So there is uniform lipid bilayer present. Now when we add with the phosphatidylcholine sphingomyelin with cholesterol, then what happens in 1:1:1? As you can see, small microdomains are actually created. So lipid phase separations occur.
So let's suppose this liposomes looks like this. Then when we introduce cholesterol, the liposomes becomes like there is small microdomains appear, and this is called lipid phase segregations. Lipid phase segregations. Basically, what happens when cholesterols are introduced, this sphingomyelin cholesterol actually binds or interacts with each other, and they cluster into small domains. That is why it is called lipid phase segregations. They segregate into more ordered and less ordered. Let's suppose these are the more ordered structure, more ordered, and rest of the lipids are less ordered. Okay. So from these two experiments, scientists hypothesized that there may be lipid rafts present in the plasma membrane. Lipid rafts means well organized or well structured lipid molecules that clusters to form microdomains. Okay.
Now there has been a long debate among the cell biologists about whether the lipids in the plasma membrane of living cells similarly segregate into specialized domains, or we call lipid rafts. Now large-scale lipid phase segregations are rarely seen in living cell membranes. Instead, specific membrane proteins and lipids are seen to concentrate in a more temporary and dynamic fashion. So this term is important. Temporary or dynamic, that means they are not stable in conditions, but temporary and dynamic fashions of these lipid rafts, facilitated by protein-protein interactions that allow the transient formation of specialized membrane regions. As you can see, this is the raft domain, hypothesized raft domain. Now in the RAP domain, the amount of cholesterol is high, this red color is high, so it intercalates with the phosphoglycerides and sphingolipids. Also, there is more glycolipids are present and GPI anchored proteins are actually present. Now, as if you minutely observe, you will find that the lipid rafts, the thickness of the lipid rafts is high, actually more than the other domain of the membrane. I have mentioned that when cholesterol actually intercalates with phosphoglycerides, it increases the thickness. And also, as these lipid rafts contain more sphingolipids like sphingomyelin and glycolipids, so the length of the fatty acids of sphingolipids is more longer and saturated. So for these two reasons, for the cholesterol intercalation with the phosphoglycerides and the presence of sphingolipids actually increase the thickness of the lipid domain. Some biochemical and microscopic evidence support the existence of lipid rafts, typically 50 nanometer in diameter. Best characterized example of raft-derived structure is caveoli. This is the electron microscopic diagram of caveoli. Now, this caveoli is rich in glycolipids, cholesterol, and GPI anchored proteins like the lipid rafts. Caveoli is involved in endocytosis.
Now the raft can be disrupted by two chemicals. One is, so lipid rafts can be disrupted by methyl beta cyclodextrin, and one antibiotic called philippine. This is an antibiotic. Now, what these two chemicals do to disrupt the lipid raft structure? Basically, they sequester. These two chemicals can sequester. That means they can absorb, sequester cholesterol from the raft domain. Now, this experiment indicates the significance of cholesterol in maintaining the integrity of the lipid rafts. So cholesterol plays an important role in the function of, or in the structure of lipid rafts in the plasma membrane. Now, one thing is that for microscopic visualization of lipid rafts, we can use one very strange molecule called cholera toxin. Now, why cholera toxin can be used to see the lipid raft domain? Because the lipid rafts are highly concentrated with lipid rafts contain high amount of glycolipids. This is important. They contain high amounts of glycolipids. Now, cholera toxin can bind with a particular glycolipids called GM1. This is actually a ganglioside. So these gangliosides are basically these are glycolipids. One example of glycolipids. I have made a separate video on sphingolipids, and in that video I have mentioned what is GM1 or gangliosides. So this gangliosides or GM1 is a glycolipids that is present abundantly in the lipid rafts. So cholera toxin actually binds with this GM1 glycolipids to enter inside the cell. So this is the real mechanism of the cholera toxin. Cholera toxin is a exotoxin. Now, to enter inside the cell, it binds with GM1, then it can enter inside the cell. So, but scientists can use this cholera toxin to visualize the lipid rafts. How? We can tag this cholera toxin with fluorescent dye. Fluorescent dye can fluoresce in a particular wavelength, so we can measure we can actually see the structure lipid rafts in the intact cells.